Okay, people, here comes another tutorial. I know you're excited.
No, God! No, God, please, no! 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 No!
Okay, yes, sorry. My favorite, ch17eukgenereg and eukaryotes. What do we need to start with? Something that we get confused about or we lose our minds, right? Every cell in an organism, in us, has all the same DNA, right? If I asked you, all cells have 46 chromosomes in humans, you would say yes, except, right, eggs and sperm. We're going to ignore those. Somatic cells all have all 46 chromosomes, which means they have all the genetic information, whether you're a brain cell or a skin cell. You have all the DNA. You have all the genes. Same. All genes. The difference in what makes a cell, their phenotype, is what genes are turned on and which genes are turned off. Gene regulation. All the genes are there. They're not always turned on. They're not all turned off. Some are on all the time. Some get turned on and off. Some are off all the time in a specific cell type, right? Liver cells make genes for liver enzymes, but they don't make genes for stomach enzymes. They don't make antibody molecules. That's a white blood cell. They don't make neurotransmitters. That's in a nerve cell. They have the genes for neurotransmitters. They have the genes to make antibody molecules, but they don't because they're turned off. Interesting. Fascinating.
No.
Okay, sorry. Proteins act in trans. DNA sites act in cis. Think of cis as sequence. It's the same on the same plane. Transacting elements are not DNA. They can move, transfer. That's where we get trans. Think about transferring them. They can go through cytoplasm. They can act on target DNA. They can act on DNA molecules, right? Transcription factors. Woo! We'll talk about it in a minute. Cis-acting elements are DNA sequences. They can only influence expression on adjacent genes on the same DNA molecule because they're part of that DNA. Like chromosomes can twist around, but they can't, jump around and go on to other chromosomes. Well, when can they do that? Meiosis, crossing over. Ooh, but I'm not talking about that. We'll talk about that. Well, actually, yeah, whatever. That's another day. That's another tutorial. Okay, moving on. Eukaryotic promoters. Remember the promoter, the generic on-off. Right? In Procs, we had an operator, a promoter, regulatory sites, all that kind of stuff. Terminator, blah, blah, blah. Eukaryotes have a promoter. They do not have an operator. Transacting proteins control transcription. Class 2 genes, RNA polymerase 2 promoters, the mRNAs. We'll just say we're going with mRNAs. We're not talking about, we didn't have time to talk about transcription of any of the other RNAs, rRNA, tRNA, siRNA, microRNAs, all that stuff. Super interesting, but we're out of time. Basal factors, the core promoter. Our favorite guy is TATA binding protein. The TATA box is a cis, right? The TATA box itself is cis. It's a sequence, TATA, and then AA or A's and T's. TAFs, TATA associated factors, TATA binding protein associated factors, a whole bunch of proteins get together to bring in RNA polymerase 2 to do complementary base pairing to what? Make RNA. So the basal factor's right there at the core. In this case, there's the TATA sequence. TBP is interacting with it, his friends. There's like hundreds of TAFs. And then a big fat RNA polymerase sitting down to complementary base pair. Proximal elements of the promoter, more than just that core, is needed. And it also helps sort of govern what's going on, like turning up and turning down rather than on and off. So up and down reg, what the heck, versus on and off. Not everything is just on off. Sometimes you need a little more, sometimes you need less. That can be a promoter thing. They are farther from the start site of transcription. Start site of transcription is the 5' UTR. The beginning of the 5' UTR is the start site. Right? Not to be confused with start of translation. This is translation only. Don't get sucked into picking that answer. The start site of transcription doesn't have a specific sequence. No spec seek. It's set up by the Tata box or other transcription factor binding sites. Blah, blah, blah. We are not memorizing this. No, it's just giving you, this is my favorite transcription factor. So these are cis acting. These are the binding sites. This guy, the octamer, is a transcription factor. He's trans. That's the protein that binds to the octamer sequence. For example, tetra. All right. So how is it really set up? Right here, if it's Tata containing, we have the core promoter, right? The transcription start site is where the mRNA starts. Somewhere in that mRNA would be a start site. Never the first 3 nukes of mRNA. What is that? Nucleotides. Nukes. This would be proximal, right? Minus 100, 200, minus 500 base pairs. All these other sites can sit there. The core promoter's in charge of basal levels, so low. If we want true expression or tissue specific or something that is actually physiologically relevant, we need proximal promoter elements. And here's some examples of the core promoter, right? We can have stuff, the Tata box. There's also initiator. If you have initiator, you usually no Tata, but sometimes they both work together. Sometimes you have neither of these. Sometimes you are Tata-less promoter. Ooh. Right? Sometimes you can have a downstream core promoter element. Sometimes you don't. TF2B recognition sites, which nobody buys those too much. Regulatory promoter, this is where all those other transcription factor binding sites go. Various combinations of core and proximal determine regulation, right? It's the key to gene regulation. This is what my lab does. My lab. Woo! Studies the proximal promoters of genes, right? What binds to the DNA? What cis-acting factors are there? That's transcription factor binding sites. What trans-acting proteins bind those sites? I don't know. I care. Do you care?
Nope.
Hey. All right, housekeeping genes, what does that mean? Something on all the time. Prokes would call it constitutive. We don't really use that word too much, but it works, so we can say constitutive. Right? Analogous to a proc constitutive. We always have to have them on in every cell, so that might be things like cytoskeleton genes to make the proteins for cytoskeleton, genes to make the proteins for ATP synthase, anything we need all the time, doesn't matter what cell you're in. There's also something called enhancer sequences, which are also cis-acting. So if we're calling it an enhancer sequence or an enhancer element, it's a cis-acting element. It's a sequence. Has some of the same transcription factor binding sites that promoters do, but they're usually far from the actual gene. And then chromatin can loop around and bring them all together and make big complexes of proteins and gets kind of crazy. But it really can turn up activity. So when you need a boost or a blast of some specific protein, you'd want it to have an enhancer. One of the best examples of that is in the immune system, our white blood cells that make antibodies when you're sick and you need to fight strep throat, for example, right? And those B cells, those white blood cells, need to turn it on, turn it up, get some serious antibody proteins pumping out there, enhance our sequences. 1,000-fold RNAs can come off of that puppy when you have an enhancer. I know, pretty crazy. So how do we think it works? Oh, it's a possible looping of DNA that brings it all together, right? So here's enhancer cis-acting. Here's a transcription factor binding protein binding to that guy. How does it work? I don't know. I like to say it tickles the RNA polymerase to hop on and go faster. The hop on, and keep going, right? As soon as polymerase in this structure gets beyond the promoter and is complementary base pairing, another polymerase can hop right on. So you can get a whole ton of RNAs if you have more and more and more RNA polymerases hopping on. So that's great. Great. Get him on there. Get him going. Another one, another one, another one. Go, go, go. So again, just going to keep going here. Sequence. So here's the promoter. The core would be this part. This part would be the proximal. Here's the core, right? That's where RNA pol 2. Ooh. RNA pol 3. RNA pol 2 binds, starts complementary base pairing, right? Then this is the start of transcription. Transcription start site, the +1 of transcription. And then somewhere, right, we're going to have an AUG. That's the +1 of translation. Then somewhere in there, you might guag. You might splice, right? And then somewhere in there, you're going to have a UGA, some sort of stop. And then you're going to have a 3' UTR. Wah! This is going to be your 5' UTR. Wah! I know, we're getting out of control here. Okay, hang on. Too much. Blah, blah, blah. We just said this. Okay, enhanceosome. Everything used to be a zome, spliceosome, enhanceosome, whole bunch of proteins, blah, blah, blah. Promotosome. No, they never said that. Okay, moving on. The transcription factors themselves, proteins, transacting. It's right in their name. They can be activator, silencer, down. So this would be upregulating. These guys would be down-regulating. Combined to the promoter, the enhancer. Silencer elements can be cis as well. These are cis. The promoter is cis. The proteins, the activators and silencer proteins are trans. Right? So turning things up, turning things down. Ooh, repressing in the case of silencers, repressors. Let's look at that. Let's look at these transcription factors and what that protein actually looks like. Quick, I'm taking too long. Again, does that, right? Enhancers, do that. Activators, turn things up. What else? Ooh, common transcription factors. We're not going to memorize what they look like, but we're going to know they have specific shape. Shape gives you function. Shape is determined by tertiary and quaternary structure. Quaternary? I don't know. Can't read my writing anyways. Structure of the protein. That is really bad. Structure of the protein. Right? Zinc finger proteins, helix, loop, helix. Right? That's this guy. Helix, loop, helix. Helix, loop, helix, loop, helix, right? And it sits in the major and minor grooves. So all transcription factors interact with double-stranded DNA, okay? The promoter is always double-stranded. Promoter. Always double-stranded. It does not open up. What opens up? The move that over here. What opens up? What opens up? Transcription start site has to open up so we can start transcribing, right? So if this is your +1 of transcription, right? This is your promoter, right? This is always going to be double-stranded. It's going to open up right here, and then we're going to get the RNA to come off. We're just knowing these are the names of some shapes. Helix, turn helix, another type, right? Here it is in the major groove, the minor groove, the major groove. Woo! We're not memorizing these. Transcription factors and transcription factors. This guy works as a dimer. So this is 2 individual proteins. Each has tertiary structure. Together, quaternary structure for function. They can't really do diddly squat by themselves. They have to be in a dimer. It's very common for transcription factors because they have to bind either both major groove or both minor groove. Okay. Again, many form dimers. Again, homodimers, 2 are the same. Heterodimers, 2 different. All dimers are quaternary structure. Again, I'm not going to ask you to memorize names. I want you to know this exists. Some other options. The leucine zipper. Why? Because there's a bunch of leucines. Again, this is a dimer. Right? And it gets together to get intercalate kind of into DNA. So it can bind there. Right? And then here's a guy that's sticking off. Woo! So exciting. Oh, zinc fingers, same idea. Look at that guy poking right in there. And definitely actually uses the metal. So we're not memorizing any of that. We're not memorizing this either. Not memorizing. But I just want you to know there's lots of different transcription factors out there, and they can bind, right? Major groove, major and backbone, adjacent major grooves, major, blah. Okay. So what? Levels of gene control right here. Compact DNA, right? So heterochrome. Shut down. Not available for transcription. We can relax it. We can alter that structure, make it euchromatin. Then we can even open it farther. We can remove the nucleosomes to make it open, right? More open, push away, remove nucleos. We make a pre-mRNA, primary transcript, heteronuclear RNA, not processed. Then we process that sucker, right? What do we do? Process, cap, splice, tail, splice some more, maybe we need to. RNA stability, how big our tail is, can help change how long we're allowed to do translation, right? We can make a protein. We can make the original protein inactive. We can do something after, post-translational, to activate it. So lots of ways to control gene expression. So hopefully that was really helpful.
Nope.
Okay, well, I did the best I can. So please be ready for, more tutorials.
No, God! No, God, please, no!